Hello, my name is Joshua Gabi. I welcome you to Fungi Mushroom. Mushrooms and toadstools form a large group of fungi which live in the soil or in rotting wood. So most of the fungi that we find in the forest, we find them in soil, and then some of them are on rotting woods. Those type of fungi are largely toadstools and mushrooms. Their mycelia spread through the soil or the dead wood, dissolving and absorbing organic substances. So what we call the mycelium is this part here. Those small hyphae, or what we would call root-like structures, are what we call the mycelium. And those ones, they spread in the soil and also in dead wood, many of them. They spread in the soil and in the dead wood. And what they are doing there, they are absorbing and dissolving organic substances. Fruiting bodies of Saliota campestris, that is a field mushroom, and Saliota arvensis, the one we call the horse mushroom, they are edible. There are some mushrooms whereby the fruiting bodies, they are edible. Like these examples, the Saliota campestris, Saliota arvensis, those are edible mushrooms. Fruiting bodies of the toadstools are mostly inedible or even poisonous. We have what we call the toadstools, a type of fungus or fungi, and this type are inedible, meaning that they are not eaten, and in most cases they are poisonous. Fruiting bodies under favorable conditions, some of the hyphae just below the soil, they mass together and form a spherical body that grows rapidly and pushes above the surface. So we have here our hyphae. Those are the hyphae. When the conditions are favorable, these hyphae, they mass together. They mass together and join together, what we are calling as massing together, and they form a spherical body. They form a spherical body, like this one that we are seeing here. They form a spherical body that grows rapidly and it pushes out of the soil. So we call it the fruiting body forming. It's the same as the fruiting body. That mass is the fruiting body. This body grows and develops three distinct regions: a stalk, a cup, and the gills. So when it grows, it is going to form three distinctive body. One is the stalk, and then the second one is the cup, the upper part, and we have the third one. Those are the gills. Those are the ones that are inside. Those are the gills. Those are the three distinct parts of the body of the fungi. First, the cup is joined all around the edge of the stalk. But later, as a result of rapid growth of the stalk and the cup breaks free, leaving a ring of tissue, the annulus, around the stalk. So when you look at the fruit here, at first it is attached at this point here. The cup is attached together here at this point, like that, all of it, without showing any gills and so on. But after some times, that one breaks away. It breaks away the attachment, and now it leaves what we are calling the annulus. The annulus here, which you see this point here, that point, the annulus, is a point left because originally it was the point which was touching this part here, the edge of the cup. The cup was attached there. That cup was originally attached to this point here. But when the stalk was growing very rapidly, the cup broke free. It broke free and it left that tissue there, a ring of tissue which we call the annulus. That tissue here is the one that we call the annulus. It is where the cup was attached. The gills are at first pink. Later they turn to dark brown and are flat sheets of tissues radiating out from the stalk and hanging vertically downwards from the underside of the cup. So here we have the gills. These ones that we are seeing are the gills. And the whole of this one is the cup. Now in the middle here, in the middle, we have the stalk. The stalk is here. All the gills are attached to the stalk. So we are saying they are flat sheets. They are flat sheets that are radiating from the stalk and they are hanging vertically. For example, if we have our cup like that, and in the middle we have the stalk. So if you draw one sheet it moves from this side, it comes and attaches itself on the stalk, and the other side it has attached on the cap. But that one goes surrounding in the middle part like this, like the way the stalk continues up. So this whole area here, the whole area here is a leaf of the sheet, that flat sheet. So when you look at it, it is hanging vertically downwards from the underside of the cap. So it is hanging down, and here it joins and it touches on the stalk. That is the flat sheet, which we call the gill. Towards the outer edge, the gills are further apart. Half length and quarter length gills are interspersed between the full length one. Now, when you look at our gills, what you are going to see is that these gills, they are of different length. You will see that we have those ones that are full length gills moving from the cap and then going and attaching themselves to the stalk. Those are the full length gills. They are attached from the outer edge, where the gills are further apart. So you can see that from this outer end, the gills are further apart. But when they are coming towards the stalk, you can see that these two gills are close together when they are coming. But as they are going out, these two gills are further apart. Now from there, we have another one which is in between them. That is the half gill. You can see that is this one which is half gill. And then you have others which are quarter gills. So that's how the gills are distributed. There are some that move from the cap to the stalk, and there are some which are halfway and others are quarter way. The surface of the gills produce millions of spores which are fired into space between the gills and fall to the ground and are carried off in the current. So we are having the gills here. Now in between those gills, there are millions of spores that are produced. Those millions of spores are in between the gills. They are fired off when there is blowing current of air. That air is going to blow them and then they will fly into the environment. So in the surface of the gills, they are the ones that produce these spores. And these ones are now stored in the space between the gills. Now, when they are kept in the spaces between the gills, they may fall to the ground, and some of them, they may be blown away by the current of wind. During 3 or 4 days, it is active life. the fruiting body of this fungus is thought to liberate about 500,000 spores per second or per minute. So if we are blowing the spores during the 3 or 4 days of its active life of the mushroom or the toadstool, it can liberate about 500,000 spores every minute. Each spore, under suitable condition, is capable of producing mycelium, which may eventually grow into a fruiting body. So when that spore falls off, it may produce mycelium, and this mycelium may end up joining together and producing a fruiting body. Thank you very much for participating in Fungi Mushroom. Please do like, share, and subscribe.